Human neurodevelopment is a unique, complex process that can cause severe disorders when things go wrong. However, how do we study the developing human brain? It is a structure that is both ethically and practically inaccessible to many of the common experimental methods that we would use otherwise in the lab. I use what's called a brain organoid, a three-dimensional stem cell-based model to study a neurodevelopmental disorder called Rett syndrome. Rett syndrome is severe, rare, and eventually fatal, with patients presenting a spectrum of symptoms that combine features of autism and epilepsy. Now, the mechanisms that cause Rett syndrome are still poorly understood, and patients often have a really variable response to the exact same medication. Therefore, there is a need to identify the specific pathways that are impacted in Rett syndrome and to approach new therapeutics in a far more targeted manner. We can derive brain organoids from patient stem cells that excellently capture and model the developing human brain, giving us the opportunity to conduct experiments that were not possible to do before. This allows us to better understand what is going on in Rett syndrome, what are the root developmental causes, as well as find and test therapeutics for new targets. Additionally, brain organoids are one of the most interesting and novel technologies to date, and they are going to be applied to a very wide variety of neurodevelopmental questions, giving us insight into both normal and disordered development.